Good morning. I'm delighted to present the nominating committee report. We have 4 items for you today. For reasons that will become obvious in just a moment, it would be highly improper for me to item number 1, so I'm going to invite Daphne Trotter to take care of that.
Good morning. It is my pleasure to introduce you to a candidate to be a new member of the MDI Bio Lab Board of Trustees. That candidate is Jennifer Benz. Jenny, would you mind standing? Since Jenny is Ed Benz's daughter, Ed has, of course, recused himself from all proceedings addressing Jenny's potential nomination, which is why I am here to recommend her to you on behalf of the nominating committee. Jenny is here because of her unique skills that are well suited to assisting the board and the lab with respect to the lab's communication efforts, a skill set that we have been hoping to increase on the board for some time, and a skill set that has become even more needed as we face these challenging times. As background, Jenny graduated from American University magna cum laude with a degree in political science, and then received from the University of North Carolina at Chapel Hill a master's degree in political science and a PhD in political science with an interdisciplinary fellowship focusing on political behavior, empirical methods, and social psychology. She is currently Senior Vice President of Public Affairs and Media Research of NORC, the National Opinion Research Center at the University of Chicago, and Director of the Associated Press NORC Center for Public Affairs Research, having been employed at NORC since 2003. As communicated in her expression of interest to the board, Jenny has deep experience leading initiatives to translate scientific and social research into accessible, actionable insights and to help organizations extend the reach and relevance of their research. Among the opportunities to assist the lab that she highlighted are to support the lab in amplifying its scientific contributions, deepening its engagement with public and policy audiences, and strengthening its fundraising efforts by clearly articulating its unique value and impacts. In light of Jenny's experience, Jan Coates, as chair of the lab's Communication and Public Affairs Committee, and I invited her to join us at a recent meeting of that committee. And she has also met with Dr. Howler and Jerry Bowers and the other members of the governance committee, other than Ed, all of whom support Jenny's candidacy. Furthermore, the Board of Trustees unanimously expressed its support for Jenny's candidacy at its May 15th board meeting. Therefore, I should like to ask for a motion for a vote of the corporation to elect Jennifer Benz to the MDI Bio Lab Board of Trustees for a 4-year term, extending through the corporation's annual meeting in 2030. May I have a motion? Second? All in favor? Any opposed? Thank you.
Well, I don't need to tell you how proud I am to have Jenny as a colleague, and appreciate your unanimous vote, so I can now withdraw my request to the Department of Homeland Security to check all the voting machines. So we have 3 additional items for the nominating committee. The first is we recommend the following slate of nominees for re-election to the Board of Trustees for a term ending with this meeting in July of 2030. Herman Holler, MD, who will, of course, be ex officio in his role as president of the institution, John A. Hayes, and Anna U. Maynard. Could I get a motion to approve those re-elections? Second? All in favor? Any opposed? Thank you. We also respectfully submit the following slate of nominees for the 2026 and 2027 nominating committee. This is my annual exercise in self-dealing where we nominate ourselves for another term. The trustee members, there are 4 trustee members and 3 members of the corporation who are not trustees who serve on the nominating committee. The trustee members up for election are Daphne W. Trotter, me, Edward Benz, Terrence C. Boylan, and John Hayes. The corporation members are Barbara Belts, MD.
You probably should take a walk with you.
and somebody else who's just been self-nominated, I guess. Jerry Bowers and Patricio Silva. Could I get a motion to approve those members of the nominating committee? Second, all in favor? Okay, thank you. And then finally, the Board of Trustees wishes to recognize the following members having fulfilled their term as trustees for their extraordinary service to the MDI Biological Laboratory by appointing them honorary trustees. Phoebe Boyer and Wistar Morris. That's for your information. We're delighted to have them continue to serve as honorary trustees. So I've served as vice chair of this Board of Trustees, I think for about 25 years now, through 2 terms of Jim Boyer's chairmanship, Peter Allen, and now have the pleasure of serving with Alan. So I think it's time for a new vice chair to take place, and I'm delighted that Daphne Trotter has agreed to be a trustee, the head of the nominating committee and vice chair of the board. I'm very honored that they've asked me to stay on the board because I'm very, very excited about the future of this lab under Herman's leadership and this incredible team that you all hear from so often. So this is to the future of the lab, and thank you for allowing me to nominate myself so many times over the past quarter century. That's my report, Alan.
And thank you, Daphne. Now we will hear from Dr. Boyd with the Treasurer's report.
Thank you. Can we get the Treasurer's report? Next slide, please. Let's see. Here we go. There we are. So as in previous years, this report to the corporation covers the calendar year 2025, and the numbers that are shown reflect the financial status as of the end of December 2025. I'll remark on the current environment at the end of the talk. The statement of financial position compares assets and liabilities. The year-end positions as of the end of 2025 are shown in the middle column and are compared with the positions of the prior year, 2024, which are at the far right. As you can see in the first line of the table, cash and certificates of deposit increased by nearly 400,000 from 2024. In the second line, receivables, including grant funding receivable, increased by about the same amount by the end of 2025 versus the previous year. Assets held in investment type accounts increased by almost $3 million over the year end 2024. This is shown in the third line. This line includes the endowment funds. This increase was mainly due to positive market fluctuations in the value of the securities held and also to major donations. The investment committee, along with the laboratory's outside investment advisor, monitors the endowment. As of June 30, 2026, which is this year, the market value of the endowment funds that are held at our investment advisor totals just under $13.9 million. For liabilities, the main category is bonds payable. The lab continues to make regular principal and interest payments on the $5 million bond that helped finance the Morris Laboratory building in 2007. The debt in its entirety is scheduled to be paid off by the year 2036. The category called Other Liabilities decreased by $360,000 at year-end 2025. Considering total assets and total liabilities, assets continued to exceed liabilities by a significant amount. Net assets were positive, and they're shown in the red box. Net assets encompass assets without donor restrictions and assets with donor restrictions. Net assets amounted to $29.9 million at the end of 2025, an increase of $4.3 million from the prior year. So of the net assets that I mentioned on the previous slide, they are distributed over 55 different categories or funds. The largest category is the capital equipment and buildings, which is shown here as plant assets in green. The general fund and unrestricted campaign donations are more flexible and are shown in blue. The other funds may be restricted by the donor by donors for specific purposes, and the institution may use all or a portion of the investment income from these restricted funds. General and unrestricted campaign funds amounted to $3.7 million at year end. And we want to take this opportunity today to remember and thank the Maine Community Foundation for their annual support of MDI Biological Laboratory by way of the Salisbury Cove Research Fund established by Tom Marin and the Marin Foundation. We greatly appreciate this ongoing support and we recognize them every year. So this slide shows the statement of activities for the year 2025. Activities include operating support and revenues and operating expenses. The numbers are broken out by those with or without donor restrictions. Total operating support and revenue in 2025 was $19 million. This amount includes grants and contracts and also philanthropic support in the form of annual giving and donations to the endowment and to board designated funds. Philanthropic support recognized in 2025 was in excess of $11 million. We express our great appreciation to everyone who contributed to the annual fund or to special gifts to the laboratory. These generous contributions are making a very appreciable difference in our ability to continue the important work being done here. Total operating expenses were nearly $17.2 million. The net of operating revenue and operating expenses resulted in an increase in net assets of just under $1.9 million from the prior year due to operations. This is greater than originally budgeted and is primarily due to the generous and timely philanthropic support that we receive. The non-operating net assets showed a positive change of $2.5 million in 2025. This reflects a combination of grants and gifts for capital purposes and positive investment returns. In summary, in 2025, total net assets from combined operating and non-operating activities increased by over $4.3 million during the year. And at year end, the laboratory had a positive net asset balance of approximately $29.9 million, as stated previously. This slide breaks out the categories of our operating expenses. Total operating expenses were $17.15 million in 2025. The majority of the expenses, amounting to 71% of all expenses, are for research and educational programs. These categories are shown here in the shades of blue. It includes $2.2 million, which are actually subawards to other institutions. Most of our subaward funding is distributed throughout the state of Maine through the INBRI program. 22% of expenses were for management and general, and it's shown here in orange. These are the costs necessary for the operation of the organization, as well as costs for administering the INBRI partnership for programs throughout the state. 7% was for fundraising activities for the purposes of obtaining contributions to support the institution. That's shown in green. This chart shows the sources of the nearly $17 million in unrestricted operating revenues available to cover our operating expenses. 40% came in the form of federal grants, contracts, and subawards, which are shown here in the two blue sectors taken together. Breaking that out, 26% of the money went to the MDI Biological Laboratory. That's the dark blue sector. And 14% or $2.2 million was distributed to the other institutions in the state through the INBRI program. That's the light blue sector. As in previous years, we highlight 2 federal grants from the National Institutes of Health that continue to provide major support to the institution. The first is the INBRI grant. This brings $1.9 million to us and $2.4 million to subawards annually. We've just begun the 3rd year of funding for the current 5-year award cycle. The INBRI grant is entering its 26th year at this institution. The other major federal grant to the laboratory is the COBRI grant. This provides annual funding to support core services and facilities like our animal model facility. We are just beginning the 3rd year of funding for a 5-year phase 3 award cycle. Philanthropy is shown in the orange and gold sectors. It's become a larger source of operating support to sustain the institution as we navigate the changing federal funding landscape. And the red sector represents our use of endowment or restricted funds, which also originate from philanthropy. We're allowed to take our earnings from those funds to use for our operating expenses. So the previous slides conclude the report to the corporation for the year 2025. The first half of 2026 has brought us additional challenges and developments, as always, and most of which are ongoing. I'd like to take a moment to summarize the situation as we see it now. Under what I've called headwinds, most of the challenges that we've confronted this year revolve around grant funding. A couple of points that I would mention. Last year at this time reported that awards for existing grant renewals were significantly delayed, which had the potential to jeopardize programs. I'm happy to report that although we experienced some delays this year, they were very minimal. However, management remains aware that potential for delays or lapses in funding continues to exist. And the other point would be the indirect cost rate. As you know, indirect costs are how grants pay for research support expenses, such as the portion of the laboratory buildings where scientists work, the utilities, maintenance, and compliance, et cetera. The lab's most recent federally negotiated indirect cost rate was 69%. There have been well-publicized discussions at the federal level to cap the indirect cost rate for all federal awardees. A mandated cap would be highly impactful if made permanent for all grants. In terms of what I would call the tailwinds that are moving us forward, we note that the many positive research and collaboration activities at the lab continue on. We again express our gratitude for the outstanding philanthropic support to our mission, which has had a huge impact on our ability to be successful in 2025 and 2026. Our faculty and program staff are seeing very real success in obtaining new grant funding. 2 of our faculty received highly regarded MIRA awards from the National Institutes of Health this spring. Foundation support has been garnered, and we have positive signs of the potential for another large grant to provide institutional program support. We await further developments in all of these areas. This concludes my remarks. Thank you very much.
Can we have a motion to approve the treasurer's report? Second? All in favor? Any opposed? Okay, with that, Dr. Schubert.
Morning. First, I'd like to thank committee members, Wistar Morris, who's on the screen, Tom, and Dean Reed for their expertise and time. The MDI Biological Laboratory Audit and Risk Committee engaged Baker, Newman, and Noyes to audit the MDI Biological Laboratory's business processes and documents. Ann Cloutier, the principal auditor, and Meredith Wicks, the audit manager, met with the committee. We learned that there was a de minimis error of classification of a laboratory expense. This minor error confirmed the thoroughness of the audit process. I thought of it as an Easter egg. And we learned that the failed process when undergoing automation had not been completed, so it's unlikely something like this will happen in the immediate future. Our audit process is thorough, and our audit shows accurate financial documentation, and we congratulate our financial staff for doing a great job.
Thank you, Dennis. Can we have a motion to approve the Audit and Risk Committee report? Second? All in favor? Any opposed? Okay, that concludes the business portion of this meeting, and we're going to adjourn that and get to, I think, really the most interesting part of the meeting. It's always a highlight of today to get an update on what's going on the research front and educational programs at the lab. So we're delighted to have Dr. Haller with us this morning. He will share some updates with us, talk about the current landscape, which is obviously a very interesting and dynamic situation, and talk about plans for the future. Herman, we are exactly on time, so I'm counting on you to keep us on time. Oh, thank you.
Thank you very much, Alan. Most likely I will disappoint you. Thank you very much for being here. Welcome, members of the board and members of the corporation. We all understand that I'm giving this report on behalf of all the staff, of all these wonderful people who are actually responsible and doing all the work. What has happened over the last couple of years? You have seen a couple of changes, like the finance report looks much better than before, looks much better because of the support of all of you. And it's a time I will talk about what we have achieved, but it's also time to look back. Wistar, do you remember this is 20 years ago that you took a shovel and you broke ground together with Terence Boylan and Dr. Forrest for the Morris Building. Only 20 years, imagine that. This is not a very long period for an institution like this. And we'll not talk a lot about the past, but thank you very much for all the support during the last 20 years. So, I will not talk a lot about the past, but I will talk about this subject that we have a dream. These people had a dream. Those of you, you can look, it's not from the 19th century. It's from 2004. And some of you can see, like Jerry Bowers, different hair, but otherwise unchanged. But this is the first year-round team in 2004. It's not easy to talk about when actually the dream that this could be an endless summer, because that was actually the dream, could be an endless summer at MDI BL. Everybody forgot about the winters and everybody were, but this is when it all started. And this is us, not right now. We are only taking another picture in a couple of weeks here at the lab. But this is the whole group, and you can see how this has grown. And now I'd like to talk about where we stand, what we have changed, and how we are going to address the future. What did we achieve? Most importantly, this is what we want to achieve. This is basically the dream. The MDI Bio Lab is a leading international biomedical research institution. This is already a lot. It's leading, it's international, utilizing the most advanced scientific technology. How bold. Artificial intelligence, when we put this together, this was before this year, this was a couple of years ago, and comparative model systems to develop new approaches for improving human health. Our campus is a gathering place for preeminent scientists, motivated students, and engaged citizens where ideas are sparked, new knowledge is discovered, critically discussed, and disseminated to the world. That's what I call a tree. And I'll tell you what we have achieved, what the reality looks like, and whether we will actually be able to get there. We have changed a couple of things. I mean, we always had a corporate structure, and we had, if I can open this, yes. And you've heard about this before, that we have an executive committee, we have regular meetings, and we try to balance this huge group of people out, and we have a new member. The Vice President of Operations is now Carol McGifford, and Carol joined us, it's hard to imagine, only a couple of weeks ago, and she already has taken over a lot. So Carola held senior leadership roles across the government, nonprofit and private sectors in DC. She's a lifelong summer resident at Mount Desert Island. She is and has strong links. She is the fifth generation granddaughter of Charles William Eliot, who was the co-founder of the park. And then she relocated to MDI full-time in January of 26. Karen, I haven't told you that, but about 10 days after you arrived on the island, it was the first time that people talked to us about, oh, she's back on the island. So I'm very happy. She has a BA in government from Wesleyan University. She has an MA in Chinese studies. She was responsible for a while for the international relationships between the US and China in several areas from Johns Hopkins. And I'm very happy that Carola is with us, and this will help us to face the challenges of the future. Welcome. Now, as always, I'll structure this into research because this is why we're all here. But I'll talk a little bit about the implications and a little bit about the strategic thoughts and the challenges which we face when we talk about research. Ian, who is sitting over there, is the Vice President of Research, and he is actually facing all these challenges, and I will just report about the thoughts behind that. So this is our faculty at the moment. But this is far from stable. I think most of you realize this. This is 26. And it has been different before, and it will change. Now, this is a terrible slide. A student showing a slide like this, we would immediately say, no, I mean, go home, come back with a better slide. However, it's a slide which has some of the complexity. Here we have the years. So it starts before 2020, and we are looking into the future after 26, and these are just the years in between. We have three colors. One is in blue, that's our faculty funding policy. This is after long discussions, finance, Jake and Claudine came up with a very structured plan how we can actually provide funding in between grants and when grants come in for our faculty. In green, this shows you the grants. Green does not mean that the blue is completely gone. Every grant has to be supported by the institution. And in yellow, we have the COPRI. This is a structural grant where we can start funding faculty in the beginning. And then with this information, you can easily see we need this money, and this is why the COBRI was so important to start the faculty, to hire people, and then they are funded, and they eventually graduate from the COBRI into the green area. It also shows you that not everybody is staying. So recruiting is one important goal, but then retaining the faculty. And they don't move to Boston or somewhere else. And there are lots of reasons to do this, but our job is to keep them here. So recruit the best faculty develop their careers and retain them. So career development is very important, and it takes years. You're not arriving here and then you publish 3 months later. You work for years on specific projects. And during this time, you have to motivate yourself. You have to motivate your students. You have to have a research group working together, because it's always, when you look at the big publications, there are 15, 20, 25 people involved in that. So it's a huge effort to develop the careers of everybody involved here and then to retain them. And you can see that we have grown. We have grown and graduated from the COPRI, and this is where we stand right now. And I'm very happy that we have more green than blue here, which you have just seen from the finance report is important. But then when we think about the future, I'm very much convinced that we need more of the yellow to go into the next generation of recruitment. And this will be an important discussion we'll have with the board later this year and over the next couple of years. So this is faculty. However, when I talk about the careers of the faculty, this also means that we have to grow the individual research group. You have just seen only the faces of the PIs, but the actual work is done by a research group, and a successful research group should consist of a principal investigator, postdocs, expensive, graduate students have to work a lot, research assistant, visiting scientists. So we are aiming for something like 6 to 10 group members. Most of you are aware that there are institutions where a research group consists of 25 and more. But I think this is a size which we can afford, and where we have the housing, the research space for that. So this is what we are planning. And therefore, we have started a graduate program to bring in more students into the labs. We're working on a postdoc program, and we still have, very important, a visiting scientist program. This is different from what it was like 20 years ago. Because 20 years ago, the scientists arrived, they moved into a lab, and they started their own project. Now, they arrive and they move, and they have to, because they need more resources, into the existing research groups and help to improve the research within these groups. So what did we achieve? The graduate program I've just mentioned, now we have to go back. The graduate program, this is, for our standards, large. We have built this over the last couple of years, and all these students are working now very hard in the labs every day to generate the science, the data, and this is for the success of the research group. But it's also a lot of obligations on our side, because when they come, they want to have a career. When they leave MDI-BL, the reason they are coming, because it's beautiful here, it's wonderful, they like everybody most of the time, but at the end of the day, they want to go home and they want to have a career in other institutions, and we have to provide this career. And when you think about it strategically, if we are not successful, if they are not successful with us, they will not come back. And the next generation will not come back, because I can tell you the network internationally for these students, which institution you go to, is very intense. And one of them, Sonia, she's somewhere, will talk about her story at MDI-BL in a couple of minutes. And this is the visiting scientist program. It was already mentioned that we are very grateful for Salisbury Cove Research Fund, because they help us to bring these visiting scientists to the campus. And in the old days, it was necessary for the summer residence program. But these days, this is a wonderful tool for our faculty and for visiting scientists that we can actually offer that. You meet one of your colleagues and you can say, well, come to MDI BL because we know we already have funding for this and to support them. I'm very happy that we have a long tradition, and some of them, like Wolfram Gößling, he's now chair of internal medicine at Yale University. He has been here a long time ago when he started his career, and now he's coming back. And this is one of the secrets of our program. We have people like Gerd Fricker, who has been coming here since 1982 as a visiting scientist. So this gives us extra stability. We have built, and you have seen this, we have talked about this before. This is the student housing. Deliberately, I'm not showing you the interior of these houses. Most of them are wonderful, not all of them. But our students are okay with that. But you can see here, we need more space for our students. At the moment, we will not grow the graduate program further, but just for the existing graduate program, we have to build more. And I'm very proud that facilities and all of us together, with the support of you, were able to renovate the inner campus. So we have now the whole, not the cottages on Spruce Point and not down at Hamilton Point. There are still things to do, otherwise it would be boring. But the inner campus is fully renovated. And this year, Ondine Cleaver, for instance, her husband, they were coming back after 12 years, and Wolfram is back, and they said, what? Wow, we have a very different memory of these cottages. They are still managed, we still keep the tradition, but they have been modernized. And then for retaining our scientists, you have just heard this, the best technology available, that's a challenge. This is why we have the course, to provide our scientists with these animal models, and the crew in the basement is working very hard to keep these animals happy, alive, and breed them. I think Sonia will mention this a little bit about her fish, how dangerous it is when they do not breed, or they are not happy, or they don't get the right food. This is what they are doing, and I'm very proud that we have established new models here, which keeps us really with a competitive edge in international research. And then our microscopes, and we have talked about this before, that we have now a wonderful microscope facility. We have excellent training there, and we have the most advanced microscopes here, custom-built microscopes. And then last but not least, these days, comparative genomics and data science, which is also something which is changing a lot. And we may talk about this afterwards. And these are the three cores we are providing our scientists with. We could do more. Perhaps when we apply for a new COPRI, we have to think about another core. And you can imagine there are other things out there which we would like to provide our scientists with. So for instance, Yannick would love to have a core here for sugar structure, because that's not in here. And there are other things which we could and would like to establish. Here you see the development of under Joel's leadership of the core. AI and the further development does not mean that we need more and more people. This is very interesting because, as you know, AI, these modern tools, they replace people at the same time. But what it actually means is that we have to grow, and Joel has to educate them, that we get the newest tools, and then we apply the newest tools. And this is where they are doing a great job. We will build. For our cores, we have to expand. Some of you have seen our microscopes in the Morris building in the basement. And I'm very proud to announce that we were able, with Cherry's help, we were able to get money from the state to renovate this area here. Some of you know that we had sharks there. Actually, we closed down the sharks because the students were swimming with the sharks at night. And but here we will have a two-story building which will be connected to Smith. And on the ground floor, we'll have our microscopes, and we'll have microscopes in the basement of Smith. And on the second floor, we will have AI. And we combine this to do something which is called digital imaging, where you actually use AI to produce better 3D pictures, etc. So we have this money in hand, and while it took us a while to build all the other buildings, this building, we will start with that, let's hope, beginning of 27, end of 26. And you'll see that for the first time ever, we'll have two buildings renovated or built at the same time. The collaborations with Maine, I've mentioned this throughout the recent years when we talked about that. But every year this is growing. And I'm very proud that Kobe has built, and they are very proud of that, a computing center in Waterville. Just for the future, because they need all these computers. But at the moment, they have too many. We are the first. Joel Graber has started this week this collaboration that we are using this computer space for our purposes, which links us much closer to Kobe. And when they develop their new bioengineering program, we'll be part of this as well. And I'll talk about collaborations with University of Maine in a moment. Now, faculty, research, the course, in which direction are we going to develop our research? What are the programs? Everybody has to interact because we are a small institution. So you know that we are doing regeneration. These are the groups who are mostly, all of us are in some way involved with that. So our animals, this is one big theme. And then aging. When we started aging at MDI-BL, it was mostly C. elegans. This is what was the first plan. And now we have introduced these African turquoise killifish, and now we have the same goal where regeneration already is. We are an internationally renowned center of regeneration. And we hope to be an internationally renowned center for aging in the future. We do environmental biology, and then we have grouped together, or they have grouped together, the groups doing RNA biology, and this is an evolving theme which we can do. And I'm very much interested in microvascular research. So developing these research areas and to make the world understand with our publications and with our meetings that we are a center of these research topics. And this is what we do also through meetings. This is a meeting Brian Moravala and Ian and Roman organized over the last 2 weeks. And I'm showing you this because all these speakers are top-notch. We had for 10 days the elite of developmental biology at MDI-BL. We all went, we all listened to these lectures. Most of these lectures, Maren, was as full as it is now, because otherwise you have to travel. And they stayed, some of them for the whole course. We had wonderful students, a full program where we educate these students with our animal models, and this is spreading the word. We are writing reports on these meetings, we are publishing reports on these meetings, and this small place on the outskirts of the living world is really the center for this type of research. And we are trying to do the same for the killifish, for aging, much smaller, but nonetheless, we are having now the second national meeting for ATK on our campus in October, and we want to grow from there, and we work very hard to make on a national basis, everybody understand that we are one of the centers. And last but not least, microvascular research, which is very dear to my heart. So I'm doing, or we are doing, microcirculation. The kidney, Romain Madeleine is doing retinal microcirculation. Some of you have heard Anindita talk about that. But now we are working together with the University of Maine because their Dean of Research and the head of the engineering department is Giovanna Guidoboni. And she's also very much interested in microvascular research, but she's a mathematician. She comes into this world as a mathematician with AI. She comes from the Eye Institute at the NIH, and they have very close collaboration. And this is what we need, and she needs us. So she has sent this year for the first time students, the best students from the engineering department to MDI BL during the summer in order to start these collaborations. And this is small, it's tiny, but I hope that this will grow as well. And then translational research. We have talked about this last year and the year before. This is becoming more important. We are interested in translational research because this is how we make an impact in biomedicine. We are aware at the Institute about this fragile balance between basic research and translational research. It's fragile because when you ask these questions in basic research, you have to blank out whether this is of any relevance in terms of money, etc. This is basic research. This is more pure. But then translation research means that you look into the possibilities, and yes, all of a sudden there are words like market, and we have to balance this, and this is what we're doing here. And we are very proud. The Neil building has been fully renovated, and we did this in record time. And we have brand new labs here. We have raised the roof a little bit, so we have offices here because office space is what we need. And we have renovated this 2 years ago, and together we have now animals for translational research, and we move in here. We got the money because we wrote these applications for research/translational research. And this is why we got the money for the translational research. But as I said, we balance this out at MDI Bio. And this is what we do here. We have this this collaboration with the Bigelow Lab, with the National Center for Marine Algae. And I could talk now for the next 2 hours about algae because I'm learning all the time about these fascinating plants, animals, algae are something in their own right, and how this works, that they are have bacteria in them and wonderful substances. This is what we are doing together. And we have a rather advanced program, so we use our model systems. We have generated all these disease models for inflammation, sepsis, complement activation, and then we treat these animals in order to prevent the disease. And then we use AI, gene expression profiling and computational analysis to find out which systems are actually affected. And then we do the next step and we go into human tissue in an organ on a chip, and we perfuse human tissue with the same substances. And then we do the same with human tissue, and then we can tell whether the same mechanisms which we have affected in our model organisms are actually affected in humans. And this has led to our first identification of a novel anti-inflammatory substance in these algae. So the substance heals inflammation in the fish, but more importantly, in human tissue, when we induce inflammation, We perfuse it with this novel substance and inflammation goes down to almost nil. So we'll find out what the substance is and we'll work on that, but this is what we'll do in NEAL. Whoop. And then always last but not least are our educational efforts. We're doing all this to educate the next generation. And I've already talked about the graduate program, and Hannah and Andrea are responsible for these students, and they do a very good job. We have talked about INBRI and Jim Kaufman and Dustin are taking care of that. And I've shown you the time course of our faculty. Jim Kaufman will retire this year. Imagine retiring. So we are retiring for the first time a member of our faculty, of the new faculty. This tells you what's happening here. And there will be more retirements in the coming years. This is why it's so important to think about the next generation. We have to think in cycles of that. So Tim, thank you very much for all your work and effort here when you first came here. We have talked about our conferences and outreach and communication. That's more difficult because Liz Marnick, this is also retaining and moving somewhere else. Liz has made a very nice career coming from MDI BL. She's now responsible on a national basis for outreach, and now we have to restructure this, and this is what we're doing at the moment. But I'd like to finish with something which is also very dear to my heart. This is this small, tiny thing here, arts meets science. I really want to build on the basis of what we have been doing with our donors and the lectures we had here, to build a program, a structured program for graduate students and undergraduates, visualizing knowledge, the art of communication. I think this is timely, not only because for me it's nice to talk about paintings, but I think it's important for our scientists, for the young scientists, to understand the historical implications. What was it like before? How do you communicate this so that you get a historical perspective on where you are and where you will go from now? And I think especially these days, this is very important. So we are writing this up. We are trying to get support for that. We want this to be a funded program in the future in addition to our educational effort. And for that, we're building, that will be the second building, which will go up next year. Badly needed. Most of you have always been asking the question, what's happening to Bowen? And every year you come in and it's falling apart more and more. The money is here. We have already made plans. And this is what it will look like, perhaps not next year when we meet here for the corporation meeting, but later in the summer, I'm very sure. And we have great plans. So it will be a welcome center. It will be for educational purposes. And then you can see with NEAL going up, with the new building for microscopy and AI, with a new building for education. Some of you, when you think about this strategically, there's only one building left, and Jerry is already working hard on that. This is the Smith building. Has been here forever, but this needs renovation as well, and hopefully we'll get federal support for that. And with that, we're building the dream. We have a dream, but we're building the dream, and when you go through this, a leading international biomedical research institution with a competitive and successful faculty and 13 to 15 research groups, this is all blue for the future. Graduate students and visiting scientists, scientific technology and microscopy, where we've already reached an international level, and a campus with new buildings and renovated cottages in order to live our dream. Thank you very much for your attention. And now I'm very proud to announce Sonia. Sonia is one of the first graduate students who came with the new program. Sonia is from India. She got her bachelor's degree from Punjab University in Chandigarh, India, and then a master's degree from the National Institute of Technology in Rourkela in India, and then she applied to the program. She came through the Hanover program, but she spent the last 5 years here at MDI Bio. It took her a while because in between the animals weren't working that well, and she was actually responsible for the transfer from the Eric Rogers lab into my lab, and she did a wonderful job. And now she graduated from MDI Bio Lab this year, summa cum laude. And she will start as a postdoc at the Mayo Clinic working on aging fish 1st of August. So Sonia, this will be your last talk for now. We'll invite you back when you have your first publications from the Mayo Clinic. And where are you? Oh. Thank you very much.
Oh, not controlling it. Yeah.
We put his right into yours.
Okay.
Here you go.
Good morning, everyone, and thank you for being here, and thanks, Raman, for your kind introduction. So today I'm going to be talking about my PhD project that I've completed here, and I'm going to talk about the 6 specific effects of intermittent fasting on muscle health and muscle aging. So the good news for all of us is that the global average life expectancy of humans have increased in the past century. It was only 32 years in 1900, and it's more than 73 years now. And it's only projected to keep increasing in the coming years as well. Now, the only sad news is that this increased life expectancy is not being accompanied by increased health expectancy. Now, because with aging comes the burden of age-associated problems, such as cardiovascular diseases, cancer, diabetes, frailty, neurodegenerative diseases, and skeletal muscle decline. Skeletal muscle decline is one of the first signs of aging that we noticed in ourselves as well. And we also lose the sense of independence as it progresses. Now, this loss of muscle mass with age is termed as sarcopenia. Dr. Rosenberg proposed this term in 1989, and this was defined as a progressive age-associated loss of skeletal muscle mass, which was further developed by European groups and also extended this explanation to decline in strength as well as physical function. Some of you might know him since he was in Tufts University throughout his life working in nutrition department. Now, after the age of 30 years, we lose 1 to 2% muscle mass every year. And this can increase to 10 to 12% once you turn 70. Now, this is scary, and this ratio and this percentage of decline that we experience every year can also increase and vary depending on where you live, and also, surprisingly, and maybe not so, whether you are a male or a female. Now, there is more muscle mass as well as muscle strength in men as compared to women. And this rate of aging of this muscle also is different for both men and women. But nevertheless, both men and women experience muscle aging. And it's sad that we still don't have any FDA-approved drug available for the treatment of sarcopenia. And US alone is spending more than $18 billion in healthcare expenses for treating sarcopenia. Now, if we look at skeletal muscle, it's a complex ecosystem of all these cell types. So there are satellite cells which are there for regeneration and repair. There are muscle fibers which are there for force generation, immune cells for protection, mitochondria for ATP production, neurons which are there for innervation and neuromuscular transmissions, as well as endothelial cells for oxygen carrying and nutrition transport. Now, a lot of coordination and interaction of these cell types is necessary for maintaining the muscle homeostasis. Now, with aging, what happens is this homeostasis is disrupted. And what we see is it manifests as increased inflammation, imbalance of protein turnover, and increased ROS production and dysfunctional mitochondria. And all of this together can lead to sarcopenia. Now, to study sarcopenia, there are several models, aging models, which are available. These known canonical models, such as mice, zebrafish, and monkey, they're living really long, like such as zebrafish lives up to 5 years. And monkeys, as we know, can live up to more than 40 years plus. And so this makes it a little bit harder for us to kind of do the aging studies with these models. And this is when African turquoise killifish comes into picture and comes very handy for the aging field. Now, this fish lives only for 6 to 8 months maximum, depending on their strain. And this is a vertebrate animal which is sharing their lifespan just like the invertebrates. Like 6 to 8 months is really small, and this is the shortest living vertebrate that we know in captivity. Also, this shows sexual dimorphism, which means that the male and female can be told separately from each other. Also, this fish beautifully recapitulates aging features, such as you can see the color loss, you can see wound healing failure with aging in the fish. We can also see tissue homeostasis failure, as well as kyphosis, which is the bending of the spine, and muscle loss. Now, a little bit about my first experience with the fish and how it all began. So I arrived here at MDI BL in the dead winter of 2021 with only fish to focus on. And I focused, and for 6 months, I couldn't even figure out how to breed this fish because whatever was published was not working. So the fish were not breeding. And after 1 year, I realized that the fish are also living for more than 10 months. And the whole point of me starting the study was because they were supposed to be shorter living, 6 months, 8 months at max, but they were 10 months. And I had no clue why they were not reproducing and why I'm not seeing what I was supposed to see according to the text. So until after 8 or 10 months into the program, I realized maybe we were unintentionally putting the fish on dietary restriction. And dietary restriction is known to increase the lifespan across species. So what if I was accidentally dietary restricting my animals? And maybe that's where they were not reproducing, and that's where they were living longer. And so I got very interested in figuring that out. And so I asked the question, can dietary restriction or the fasting can produce this longevity phenotype in the fish? And this became my PhD project, where I got interested in asking how does dietary restriction influence lifespan, and what is the skeletal muscle aging looking like under this condition in this fish? So towards this goal, we started with the optimization of dietary restriction protocol. And my choice of protocol was going with intermittent fasting, since it was easier, that I don't have to every day manipulate how much to give. So for ad libitum conditions, we gave the fish throughout its life the maximum amount of food that you can give in a day. For IF1 protocol, which is intermittent fasting 1 protocol, there was 2 days of fasting, 1 day of feeding. and so on. And for intermittent fasting method 2 that we tested, there was 4 days of fasting followed by 3 days of feeding. And then we look at what happens to their lifespan and what happens to their reproduction. Now, I was very surprised to see that intermittent fasting method 1 was the one where we see an increase in the median lifespan of these animals. Also, in both IF-treated animals, the dietary restricted 1 animals, there was a decrease in reproduction, similar to what I was also seeing in my fish when I initially came. So IF1 became the important and most interesting phenotype for me, and that's what I used in my next studies. So I tested for both male and female as well, that IF1 increased the lifespan of the females by 21% and for the males by 53%. Now, what was also interesting was that these animals which were put on diet restriction, this old IF-treated animals, were also smaller in size, and both in terms of their body weight and body length. So what we see here is that with IF treatment, there is an increase in lifespan, but there is decline in growth as well as reproduction of the animals, which is also kind of is the trade-off that is associated with the, explained by a little bit by soma theory. So now we got interested in asking, we see this increased lifespan, right? Now, does this also translate to health span? Is my animal like just living healthy or just living longer? So for this, we tested the swim capacity of the fish. And the way we do it is we put the fish in the swim tunnel, increase and decrease the flow of the water, and then check if what the swim capacity is. We noticed that after a certain point, when they're exhausted, they can no longer swim. And that's the maximum swim capacity that we notice. So with age, what we notice in both male and female is that there is a decline in the swim capacity of this fish. And with our IF treatment, both male and female bounces back. So at the same age, but with IF treatment in the older age of these animals, there was an improved swim performance. So IF improves the swim performance in this fish. So now we are going to investigate why is this? Now, why are we seeing this phenotype? Now, first thing that comes to our mind is maybe muscle mass index is higher, because that's what we measure when we have to kind of diagnose somebody with sarcopenia in the clinics. So we looked at muscle mass index of this fish using the micro CT scan. And to our surprise, there was actually an increase in the muscle mass of the males. And there was no change for females with age. However, with our treatment, there was decline in this muscle mass index for females. So there is not a correlation that's there. And so basically, higher muscle mass is not translating into this better swim capacity that we see. So higher muscle mass index here does not correlate with better swim performance. Then the second thing that we want to check was, what about myofibers? What if the count of these fibers is increasing? And maybe the overall muscle mass doesn't change, the count changes, and maybe the diameter as well changes. So we checked the count and the size of these fibers. And to look at that, we established segmentation of these myotomes. And using elastic, and Hannah here helped with establishing this imaging pipeline, we found that there were, with aging, in case of males, there is both hyperplasia and hypertrophy. So quickly, what hyperplasia and hypertrophy is, if the size of this fiber or the diameter of this fiber increases, we call it hypertrophy. And if only the count of this fiber increases, we call it hyperplasia. So what we see in case of males here with aging is that there is both hyperplasia and hypertrophy that is happening. and there is only mild hypertrophy that is happening in case of females, which is still baffling because this does not translate into why their performance declined. Now, with the IF treatment of the same age, what we see is that there is neither hyperplasia nor hypertrophy that is happening in case of either male or females. So we have male and females using different cellular mechanisms for muscle growth during aging, as well as IF attenuating this dimorphic hyperplasia and hypertrophic response during the aging. So remember, we had this slide where I showed that there are different cell types which are present in the muscle, and this homeostasis and this talk between them is required to maintain this homeostasis. So now we were interested in finding out what if that is hampered. Now to look at that, we would have to define what kind of cell types are present in this muscle type of this killifish, since that has never been defined before. So we resorted to single nucleotide transcriptomics analysis and identified 11 different cell types which are present in case of the muscle of this tissue, of this fish. So we had endothelial cells, fibroblasts, oligodendrocytes, neurons, as well as macrophages, which we could identify. And on the subclustering of this myonuclei, we found several different fast and slow types fibers which were present in the fish. Now, what was also interesting was that there were no specific cell types which were present with age, sex, or even IF treatment. So basically, all these treatments and both male and female have same types of cell types in their muscle tissue. But what was different is the proportion of those cell types. Now, when we looked at the myofiber composition, we saw that there is a muscle fiber plasticity that is happening. Here, in case of the females, there is a decline in the fast fiber types, which is from here to here, and there is improvement in the measurement of these fiber types in red. So this was supposed to be this whole panel till pink. So, and we see this phenotype only in case of females. So there is a decline in fast fiber type population and with age, which goes back with our treatment. And that happens only in case of females and not in males. And this is very similar to what we also see, not the sex-specific part, but what we see with the decline of fast-twitch fibers with age in the fish, in humans as well. So with age, fast fiber type declines and IF preserves it. And there is also a gene expression profile, which is looking very similar between this gray and the red, which are always lying towards the same side. So there are V or A-shaped curves which are being formed when we look at these gene expression. And that's true for also the other cell types that are being present. Now, when we look at the cell-to-cell communication data using the cell chat analysis, here you're looking at a circle plot. So each of these dots represents one cell type. The darker this line is between them, the stronger is the communication. And so what we see here is that when we go from young to old animals, there is a decline in the number of these interactions. And this is true for both male and female. So there is a decline in the number as well as strength of these interactions in both male and female. And I have partially or not at all in case of males reverses this, or improves this, I should say. Now, what we can also look at using the cell chat analysis is that there is these patterns of information that flows between these cell types, right? So this is an outgoing signal, this is an incoming signal towards the secreting and the targeting cells. And this cell chat analysis also helps us to know what the putative cell-to-cell signaling this could be. So to summarize this, what we found was there was sex-specific cell signaling pathways which were regulated by aging as well as maintained under IF. So in case of females, there were more of axonal guidance pathways that we saw, and more angiogenesis, as well as increased IGF signaling, which is the known pro-longevity pathway, which shows up under the IF treatment in case of females, but not in males. However, in males, what we see is with IF treatment, we see that there is a decline in the fibrotic genes, as well as increased angiopoietin-like signaling, which kind of regulates lipid metabolism. So you can appreciate here that there are a lot of metabolic differences that exist between male and female, as well as the pathways that show up with the IF treatment. So to summarize this, what we see here is that there is increased lifespan in case of both male and female. We see a trade-off of growth and reproduction in case of both male and female. And there is a associated improved swim performance, which is also observed in both male and female. There are some molecular signatures associated with that from our bulk transcriptomics data, which I didn't show here today, which shows that there is improved translation as well as myofibril organization, which is accompanying this. So the end result of this is that increased lifespan is followed up by improved swim performance in both male and female. But how they're doing it is they're following different trajectories. So this distinct trajectory looks like for females is that there is a decrease in axonal guidance pathways and fast twitch fiber switch, which is being improved by the IF. And while in case of males, it's more about increased inflammation and ECM remodeling that shows up as a part. So the take-home message for this would be that even if the end result, which is the increased lifespan and increased health span, is the same, both male and female follow a distinct trajectory. So that's why sex is an important factor to consider when we are designing therapeutics for muscle aging. And also, killifish is a really great model to study the sexual dimorphism in muscle aging. So with this, I would like to thank my lab and thank my supervisor for giving me this opportunity to work on this project. And also, especially Hannah Summers for helping with the imaging data, and Joel and his whole team for the work on bioinformatics. And also the comparative animal model, Eddie and the team for taking really great care of the animals, as well as my funding sources here. And thank you all for being here today and giving us this opportunity to continue our work. Thank you. Do we have time?
And now we're going to hear about fundraising from Jerry.
Well, good morning, everyone. Wow, it's so lovely to see so many friendly faces out here in the audience. I think we're going to have to think about overflow seating for the annual meetings from now on, which is really lovely. I want to just spend a few minutes chatting with you this morning, giving you just a quick look at where we are in terms of the federal funding scene, but also where we are with our fundraising. And I think most importantly, give you a sense of what we're thinking about for the future. So I'm going to try to be pretty brief. I know we've kept you here for a while already this morning, and you're probably ready for a break. So we will try to go through this relatively quickly. So I don't think I have to remind anyone that we are living in kind of unprecedented times when it comes to funding for scientific research. And so I don't think any of us need necessarily a rehash of what the last 18 months have been like. But I do want to talk a little bit about some of the current challenges and really what we're doing to try to mitigate the impact of those shifts in the federal funding landscape and mitigate the impact, particularly on the work that's happening here at the MDI Bio Lab. So it has been, wow, I really messed that sequencing up. Let's see, it has been, again, a pretty volatile year. I think all of you are familiar with the current administration's continued efforts to reduce federal funding for scientific research. This year, we saw a request for a 40% increase in the NIH budget and a 57% decrease. Did I say increase? Decrease in the National Science Foundation budget. So thankfully, due to the advocacy work that continues throughout the scientific community, Congress repudiated those proposed reductions and indeed funded even a slight increase in the federal budgets of both NIH and NSF this year. So despite that, we are still seeing some major changes within these agencies in terms of how federal grants are awarded, what the process looks like for reviewing those grants. One example is that pay lines have now been eliminated. And so for those of you that aren't really familiar with the federal funding process, when your grant is reviewed by a group of your peers and it is given a score. And so based on the money available, there's sort of a cut line that is established. So anything that scores above that cut line is typically funded. Now that process has become pretty opaque. And so the pay line has been eliminated. And there is now a new innovation score, which has not been well defined in terms of how those grants are awarded. So as a scientist, you can imagine that makes it very difficult, very challenging to know sort of how to get your grant funded. If it is reviewed, what does that mean? And so even though those scores still exist, they don't have the same weight in terms of being able to anticipate whether or not your project will be funded. We had a grant that was awarded today, and I'll just give you this example. It has nothing to do with the quality of the grant. The grant wasn't even discussed at committee, yet it was funded. So it's very confusing in terms of how these things are awarded, and we're still learning, we're still trying to navigate what that means. And so from a fiscal perspective and being able to relatively accurately predict what your revenues might look like on an annual basis, that's the challenging part. So we're continuing to keep a close eye on things, really trying to read the tea leaves a bit, anticipate, trying to develop some sense of the new normal. And I think one of the most recent changes that has really caused us a fair bit of angst in looking at what this means for the future of federal funding are the uniform guidance changes that were proposed by the Office of Management and Budget most recently. So there were well over 320 proposed changes, and these don't just impact federal research grants, they impact all federal grants. So whether you're a municipality and you're looking for funding, it really doesn't matter. It's across the board. And so this is something that we have spent a fair bit of time working with our congressional delegation. I see Adam there, and thank you so much for being here today, member of Senator King's team. I just want to say how incredibly fortunate we are to have the delegation that we do. They are so responsive. We have access that is really unprecedented. They are constantly checking in with us to see, how we're feeling about things. How are these proposals impacting your work? That's really just incredibly helpful and beneficial. So I want to really express my appreciation to them for the time and energy that they are all putting into helping us navigate the current circumstances. So we put in, there was a public comment period, a very sort of abbreviated public comment period, about 45 days. I think the document itself was over 400 pages. So as you can imagine, it's sort of a lot to try and anticipate what these changes, how those changes may impact an institution. But overall, there were an estimated 500,000 submissions to that public comment process. So I think that gives you an indication of just how concerning some of these changes were. One estimate, according to AI, is that about 97% of those changes were negative, were opposed to the changes. So what does that mean? Well, I think what it does is it gives us a glimpse into, number one, the potential impact, but it also sort of lays a foundation for potential litigation to prevent some of the implementation of those suggested changes. So we're keeping an eye on that. We'll continue to monitor. There was some work, some advance in that, and I'll share that in just a minute. But we're also, of course, now looking at the 2027 appropriations process. So what does that look like given the midterms, given just some scheduling slowdowns in the Senate, and all eyes are on the midterm, and those of us here in Maine, it can't come quick enough. So it's an interesting process. But again, I really just wanted to kind of take a moment to give a shout out, both at the federal level, but also the state level. So we are very fortunate to have wonderful representatives, and I think people who are indeed supporters of the work that this institution is doing. So quickly, on the OMB side, just to give you an example of sort of how this works. So I think overall, there has been bipartisan pushback on some of these changes. We saw the House come out with 122 members signed a letter that were opposing these changes. Senate Democrats did the same, and Senator Susan Collins came out with a letter strongly opposing some of the provisions that were suggested in these changes. So again, all contributing to this sort of concern, or elevating the level of concern for how these changes would impact how science is done. I thought I had one more slide in there, but in any case, I did want to just mention that we also saw earlier this week a court ruling that may give some indication into what the potential litigation will be for some of these changes in OMB. So I think we had a case where there are 23 states that joined together to sue OMB, and currently the initial ruling is in our favor, that they would not be able to just sort of eliminate a grant contract just because the administration had changed its priorities. So it'll undoubtedly be appealed, but I think it gives you a sense of sort of how things are moving forward. So in light of that, I am not able to back up. Let's see, how is this going? Nope, just not going to back up. Sorry, hold on just a sec. Let's see, how do we? I'm pushing the wrong button, that's why. There we go. So I did want to just mention, in addition to the federal funding that we all are very familiar with in terms of how we fund research, it is also, as Dr. Haller highlighted, playing an enormous role in how we are modernizing our campus. So just since 2022, MDI BL has received $5 million in federal appropriations. So these are grants that come through as part of the appropriation process itself. As you know, Senator Collins is chair of the Appropriations Committee. And so we have been very, very successful in securing some of this funding. Dr. Haller mentioned the Halsey Lab renovation, the current Neal Lab renovation. We also have plans to establish a Center for Advancing Science Education in the rather sad looking Bowen Hall that's out there at the beginning of our campus. And then he also mentioned this new development of a model development facility, which is funded by the Maine Technology Institute, a state agency. So it's really a combination of these activities that allow us to continue to invest in the infrastructure that makes all of this science possible. And then for fiscal year 27, we do have a current request in with both Senator Collins and Senator King to renovate the Smith-Kingsley building on the lower campus, sort of the one, we didn't realize really how bad this building needed a renovation until the new Neal Lab. You're up there and looking at poor Smith, it looks a little sad. So we are very excited about what that potential grant could do to transform not only the science here, but also just the physicality of our lower campus. So with that, I want to move into just a quick glimpse at where we are with our fundraising. I think you all got a little bit of a taste of that this morning in the Treasurer's report and in Dr. Haller's report. Just really how tremendously grateful we are to the support that all of you are providing this institution, particularly in this moment of unprecedented change on the federal funding landscape. It is really what has allowed us to continue the work that we do. There's no question that without that support, it simply would not happen. So I want to really recognize that. Just to quickly show you, this is a 3-year look back, a year-to-date look back in terms of where we are with our operating fundraising, what we typically call annual fundraising. This is to support the day-to-day operations of the Institute. So as you can see, 2026 is shaping up very nicely. We currently have about $8.4 million total in cash received and in commitments that have been made thus far. And that will get us pretty close to our $10.8 million revenue goal for the year. So we're feeling good about things. Still have a ways to go, but I think overall it really shows the tremendous outpouring of support that we have been receiving, and also I think reflective of the amazing quality of work that Sonia just gave you a great example of the quality of work that is happening here, and that in and of itself is really generating a lot of interest and support for the institution. I wanted to show this slide just to remind you, several years ago, when in 2023, when we put together our strategic plan and we outlined these 4 major priorities that are included in that plan. And one was the forming of the MDI Bioscience Initiative that Dr. Haller spoke about, translational research. We also knew that we needed to invest in our world-class education and training. So you heard a bit about the graduate student program. You saw a wonderful example of the results of that program this morning. We knew we needed to modernize our campus. We have a beautiful 100-year-old campus here, but many of our buildings are almost that old as well. So they need a bit of TLC. And Dr. Haller showed you how that strategic plan, the fundraising that has gone along with that, has enabled us to renovate nearly every building on the lower campus. We still have some work to do in terms of our outlying properties, but I think in that day-to-day functioning sense, we're in pretty good shape. And that, again, is due in large part to those federal appropriation grants that we just talked about. And then most importantly, we knew we would need a significant source of revenue to continue to recruit and retain the faculty that are here. And so that's the sense of where we are now. We estimated that that would cost about $45 million. We would need to have a fundraising initiative to attain that. And surprisingly, we have done very, very well with our fundraising. Again, thanks to all of you. I would say we currently have raised $48.2 million toward that $45 million phase 1 goal. So we are feeling really fortunate to be able to say that, you know, we do now have the capacity to raise the resources that we are going to need. If there's a downside, I would say it is that things, you know, the funding model that we envisioned back in 2023 no longer really is true. And so the length of time that it takes for a faculty member to get his or her independent funding is much longer than what we had initially planned. And as a result, more expensive, right, to continue that support of that very viable work. And so we're about halfway through our strategic plan now, and we recognize that we're going to need to raise significantly more dollars to continue to support our faculty at the current level. And so I just want to take a moment to sort of digest that, because when I joined this institution 28 years ago, we were raising $62,000 a year. So this is a big leap forward, and all of you are really responsible for that. The Board of Trustees has really given the vast majority of this money. And so I am so grateful to all of you for making this possible and for giving us a very important springboard from which we can move forward. So thank you to all of you. So one of the things that we have really been working closely with the board and various committees is to figure out exactly what that next phase looks like, not only from a strategic planning perspective, but also from a fundraising perspective. So one of the things that we have learned, of course, during this process, or one of our goals during this process, has been to increase our endowment. In 2022, our entire endowment portfolio was about $7.3 million. So you can see that we have been adding to that, both through philanthropic support, but also through market performance, right? That has played a big role in our ability to grow that stable source of funding that is so essential for the institution. So one big question is, what's the right size of an endowment for an institution that has roughly a $20 million operating budget? So we've been thinking a lot about that. The general rule of thumb is that you would have a 2-to-1, so your endowment would be about twice as large as your operating budget. So that's kind of what we're aiming for and thinking about. How do we continue to support the institution's operating needs while at the same time meeting our campus infrastructure needs and now raising a significant endowment. So that's part of our challenge. That will be the task that the board and others will be focused on in the next 6 months as we kind of think about updating this strategic plan and making sure that we have the resources that we need to continue to be successful. So in thinking about that, we have some very specific goals or needs, I would say, to invest in that endowment and to ensure that the institution has that stability that it will require. So we've talked a bit about our graduate student program. Currently, there are really no viable federal sources for funding non-US citizens who are coming to the United States to do their work. So as we try to read those tea leaves, as we try to think about what the future looks like, we recognize that we are going to need about a $20 million endowment to continue to fund this program, which now costs roughly $1 million a year to operate. So this is a big need. And as you can see, the training is essential, but these students are really the engine of what drives so much of the research that's happening here. So the research and education components are deeply intertwined. And so you can't really have one without the other. So it's an essential part of what we have built here over the last few years. We also recognize we are going to continue to need to provide whether you want to call it bridge funding or pilot funding for our faculty as they are working to get these federal grants. There's no question federal grants will become more and more competitive. We're going to see shrinking budgets, I think, for the foreseeable future. So how do we weather that? How do we prepare ourselves to be able to continue to do the quality of the work that we are doing? I think, building about a $15 million endowment that will help support faculty research going forward has to be part of that equation as well. So that gets us to roughly 35 million. And if you combine that with the 15 million that we currently have, we're in that $50 million range, which I think is probably a good place for us to be, a good goal for us to be thinking about. That's a big number, but I'm confident that given the amazing support that we have received from all of you, that we'll be able to get there. It won't be overnight for sure, but I think we are making some progress, and that's really where our focus is at the moment. So in light of that, I want to share a very exciting announcement with you. I think many of you may recognize the faces in this photo. Earlier this year, we were thrilled to receive a $1 million gift to establish the Epstein-Myers Scholarship Fund. So in recognition of the education programs, the extraordinary reputation that this institution has built, I think the two folks that you see here, John Epstein and Maggie Myers, have both kind of been part of MDI BL's legacy. Their families have been part of our legacy, and they both have experienced what it's like to spend time here at MDI BL. And this quote from John, I think, really captures their feeling, their sense of wanting to preserve those opportunities for the next generation. So John and Maggie, we are so grateful to you for setting this up. Thank you. So where that leaves us, as I mentioned at the outset, one of our big challenges is to figure out how we can continue to raise money to support operations, that sort of day-to-day need, while at the same time addressing this need to grow our endowment. So we are very fortunate to be working, as I have said numerous times, within a tremendously generous group of donors. So we have one donor who has agreed anonymously to make a challenge to our community. This donor will support operations at the level of 1.5 million if we can try and raise 1.5 million to match it for endowment. So this is relatively new. We're still thinking a little bit of how it will be structured, but I wanted to give you a sense of how we might achieve this, because I think it's such a tremendous opportunity for us to do both things at the same time. So what does that look like if we want to increase our endowment by 1.5 million? Well, there are a few ways that we could tackle this. I think some easy ways that folks may not be aware of to think about how the institution can grow its endowment over time. You know, the first way is an obvious one. You can make a cash gift. So we're happy to help you do that. And we have lots of different ways that you can support our endowment. We have unrestricted endowment. We have many named funds here, many of whom you all have helped us establish in recognition of the legacy of science and tremendous educational programs that have been done here. So a lot of wonderful ways to do that. You can actually just create a bequest in your will. So it may just be a single line that you can put in your existing will to consider the lab as a recipient of your estate. You can designate us as a recipient of a retirement account, for example. Those are one of the most heavily taxed accounts that you have, one of your heavily taxed assets. So it's a wonderful way to minimize tax liability on your heirs. Maybe you have a life insurance policy that you had years ago when your children are small and maybe no longer have the same needs for that. You can name the laboratory a beneficiary of that life insurance policy. Some of you age 70 and older may know that you have a mandatory qualified charitable deduction available to you through use of your IRA, your retirement account. And there are also opportunities for those over age 65 to make a charitable gift annuity. You can continue to receive income, and then at the time of your death, the residual of that would come to MDIBL. So lots and lots of different ways that don't necessarily require involvement of an attorney or a financial planner, but that can be done in a relatively easy way. So we would love to invite all of you to think a little bit about that. What are the ways that you can continue to support the institution on an annual basis, but also help us plan for the future by thinking about how you could include the lab in your estate plan. I think it's an exciting opportunity for MDIBL. I am incredibly grateful to the generosity of our donors for helping us think through these challenging times and envision, as Dr. Haller said, what the dream looks like. How do we get there? And I'm confident that we'll be able to do it together. I think this is a wonderful, wonderful time for MDIBL, and I just want to express my appreciation to all of you today. Thank you so much.
Jerry, thank you very much. Sonia, I think everyone has a very clear idea of both the progress the lab has made, the exciting work that's being done, the exciting future that's being planned, but also the very significant challenges, particularly given what the federal funding scene looks like and is likely to look like in the future. So I also just want, and Herman, we are on time, so thank you. I want to thank you for being here in person. Thank you for being here by Zoom. Thank you for your devotion and interest and commitment to the lab. Obviously, we could not do what we do without your incredible support. And you know, your support has been amazing so far, and we will continue to need it in the years to come. So thank you so much. Let me just mention that everyone is invited to lunch in the tent outside. There also will be tours after lunch of the renovated Neal Laboratory for those of you who would like to do that. And with that, I would entertain a motion to adjourn the meeting. Second? All in favor? Thank you so much.